Now, some of the receptors are going to be associated with our cells are referred to as toll-like receptors. These toll-like receptors are thought to be evolutionary and ancient receptor class because of their structure, because they're found across so many different animals, not just mammalian, but you find them insect, arthropod, whatever, that we think that this is one of the original. May not be the original, but we think it dates back quite close to. We find that the toll-like receptors are homologous. They are highly conserved across animal, the kingdom Animalia. We find that depending upon which animal it's in, there's going to be a whole host of different defense mechanisms, depending upon the host, depending on the animal, also depending upon which cell type. We find that in us humans, there are 10 different classes of toll-like receptors. Mice have 13. It's going to be a variable number between the animals. Depending on, no matter which class it is, what type it is, we see that they recognize specific PAMPs. Remember, PAMPs, pathogen-associated molecular patterns. Specifically things like lipoteichoic acid. This is a specific lipid protein moiety that is in gram-positive cells that holds together their thick cell wall. Also, different toll-like receptors will bind to lipopolysaccharide. This is a sugar protein lipid that is found in the outer membrane of gram-negative bacterial. We find that while majority of them are going to be associated out or on or near the plasma membrane of our eukaryotic cells, some of them are going to be on the endosome. Leucine-rich repeat is going to be one that associates with the endosome. This one's looking more for for viral proteins, because remember, since this is going to be on the endosome, it's going to be inside the cell facing into the cytoplasm. Only thing it's going to come into contact with will be certain viral proteins. So when you look at the different toll-like receptors that you're going to find in most mammals, they're going to have different ligands. Remember, a ligand is what binds to a receptor. It activates a receptor. The ligand can be very general, or it can be highly specific. TLR1, 2, heterodimer, certain bacteria, proteins, lipoproteins, liposugars. Going to find these monocytes, dendritic cells, mast cells, TLR2, TLR6, heterodimer, the same thing. Looking for beta-glucans, some bacteria, most of the fungi. TLR3, this one is going to be natural killer cells, and it responds to the double-stranded RNA that you're going to find in viruses. Got to remember, us mammalian cells, us eukaryotes, we don't do double-stranded RNA. We may have certain regions of our messenger RNA and our tRNA that fold in on themselves, but it's very small, and messenger RNA is short-lived anyways. TLR4 goes for LPS, lipotoic acids, flagellin, single-stranded. So you see there are different ligands found different cells. Some of the cells have multiple TLRs. Look at the macrophages. The dendritic cells, that's their job. They're looking for a host of things, not you. Other ones are going to be highly specific for which, TLRs they want. When you look at them, many of them are either going to be singular, or they're going to be double. They need to be bound to heterodimer. That's what dimer means. Individual TLR2 does nothing. Individual TLR1 or 6 does nothing. It's only when it's bound to 2 will 1 or 6 do something. Will it be able to recognize specific ligands, whether it be diacyl lipopeptides or triacyl lipopeptides? Now, when it comes to PAMPs, here they're showing a lipopeptide. It's a amino acid polypeptide that has a lipid attachment. So this means it was in the membrane of a bacteria. It got released somehow, and TLR2, TLR1 don't always associate with each other. They associate with each other only when they are both bind to the lipopeptide. When they are both bound to the lipopeptide, what you see here is the cytoplasmic domains will now come to contact. 1/2, 1/2 does nothing. Together, they form an active protein. That active protein has an enzymatic activity. Here we see a computer-generated ribbon model showing you how all of this fits in to the cytoplasmic membrane. Different view showing the receptor portion. Now, once the toll-like receptor, whether it's a heterodimer or a singular, once it binds its ligand and its cytoplasmic tail becomes activated, we see that there are different pathways depending upon which toll-like receptor gets activated. Which ones, how many? They have what's referred to as the NF-kappa B. NF-kappa B, as well as MAP kinases, mitogen activated protein kinase, MAPKs. They will also get activated. Interferon regulatory factor, IRF, as well as AP1. All of these have different components within their pathways that will determine, depending on the cell, what its cellular response will be. Ultimately, one of the responses is going to be the expression, the making of cytokines and chemokines that will then be released by the cell. Some of the cytokines are also going to be known as interferons. Interferons, based on their name, interfere with what's going on. So when you look at it, this whole host of things going on. You get the dimerization here, TLR1, TLR2. Their tails come together, and then you get this complex assembled. The MAL and the MIDE D88s will only associate if the tails are associated. The MAL and the MIDE D88s associating together like this stably will recruit in IRAK1, IRAK4. IRAK1, IRAK4, again, associating like this, brings in E3 and E2 ligase, also known as TRAF6 and TRICA1. All of this together brings in other more and more ubiquitization of TRAF6, TRICA1 by TAC1, which can only interact with TRAF6 if it's bound here, causes all of this. TAC1, TAB1 and 2 add ubiquitin to TRAF6, which starts this whole process of breaking down I kappa kappa gamma, also known as NEMO. All these go together and then activate NF-kappa B. NF-kappa B has this regular I-kappa B. I-kappa B bound to NF-kappa B. NF-kappa B is also known as P65P50. I-kappa B, all of this, which is due based on TRAF6, tricycle 1. All of this degrades I-kappa B, allowing NF-kappa B to be activated, and then it goes finds a promoter. Turns out NF-kappa B has a high affinity for specific DNA sequences. Those DNA sequences happen to be cytokine genes. Once it upregulates the production of the cytokine genes, some of the other genes that will also be upregulated, one of them happens to be I-kappa B. So it increases the expression of the cytokines as well as I-kappa B, which will then bind to NF-kappa B, which will be transported back out to the cytoplasm. End of expression. Now, one of the things I just mentioned is interferons. Interferons are a type of cytokine that will also be upregulated, expressed, and sent out into the surrounding interstitial fluids of that cell that's been activated. You go through and you see here again TLRs, dimerized, the ones that have to dimerize. You get the tails, you get different pathways. No, I'm not going to expect you to memorize the different pathways. You get, finally, some form of a promoter trafficked into the nucleus, and you get expression. Interferon beta, interferon alpha. Turns out whether it's interferon alpha, interferon beta, doesn't matter. Interferons are antiviral. They will tell different cells to react in specific ways. If there's a chance that that cell is infected, to die. To increase its expression of MHC class 1. So that way, CD8 cells will know which cell to come and tell to die. Interferons are also now being seen as a great antiviral for double-stranded RNA viruses. So it's being looked at as a means, short-term, for treatment of different viral infections.